Hello students, let's study meiosis today. Okay, now in meiosis is the type of cell division in which a single cell gives rise to four daughter cells. How it gives rise to four daughter cells? Suppose this is first cell, it undergoes division, first division, it forms two daughter cells. These two daughter cells again divide and results into the formation of total four daughter cells. Means from one daughter cells, four daughter cells are formed. It is completed in two stages. First is called as meiosis 1 and another one is called as meiosis 2. Okay. Meiosis 1 is called as reductional division. Meiosis 1 is also called as reductional division because the number of chromosome get reduced in daughter cell. Suppose if mother is having 46 chromosome, the daughters are having reduced number of chromosome, that is 23. This cell again undergo division and it results into the formation of four daughter cell. Now this M2 means second meiosis division is called as equational division. Meiosis 2 is also called as equational division because the number of chromosome remains same in mother and in daughter cell. Means if the mother is having 23 chromosome, here the daughter will also have 23 chromosomes. So that's why it is called as equational division. And meiosis always takes place in reproductive cell, okay, in gametes. For example, in sperms and in ova, these meiosis takes place. That is, meiosis takes place only in reproductive cells. Now, first starting with meiosis 1. Just now I told you meiosis is completed in two stages, meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So here we are studying, first we are starting with meiosis 1. Now, meiosis 1 is completed in four phases. Meiosis 1 is completed in four phases. Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. Okay, but this prophase 1 is lengthy step. It is a lengthy process. That's why this prophase 1 is again divided into 5 subphases. That is liptoten, zygote, pachytene, diplotin, dikinesis. Again I am repeating, we are studying meiosis. Meiosis is completed in two phases, meiosis 1 and meiosis 2. First we are starting with meiosis 1. Meiosis 1 is subdivided into four stages. Prophase 1, Metaphase 1, Anaphase 1, Telophase 1. This prophase 1 is the lengthy process and it is again divided into 5 phases. Liptoten, zygote, pachytene, diplotin, dikinesis. So let's start with prophase 1 first. Prophase 1, and we are studying here first step of prophase 1, that is liptoten. See, now what happens here? In liptoten, the chromatin network get condensed. Okay, here what happens? Chromatin network means the DNA and the histone protein molecules, whatever present inside the nucleus, that is called as chromatin network. This is called as chromatin network. What happens here? The chromatin network get condensed and the chromosome appears to be long and thin. Just see the size of chromosome. Chromosome appears to be long and thin. Now, what about centrosome? This is centrosome. Centrosome, which is already duplicated in S phase of interphase. From one centrosome, two centrosomes are already formed in S phase. These two centrosomes start migrating towards the opposite pole. Only two things are taking place over here. Here the chromatin network gets condensed and the chromosome appears to be long and thin. And second point, the centrosome which are already duplicated in S phase of interphase, they start moving or migrating towards the opposite pole of the cell. So here we have completed about liptoten. Now coming to zygote. It is the next stage of prophase 1. In zygote, the homologous chromosome. Can you see different types of chromosome over here? One is from maternal side and another one is from paternal side. These two chromosomes, homologous chromosome, form pairing. They form homologous chromosome, form pairing here. Then what happens? why they are remain close to each other or why they are in contact with each other? Actually, no, they are bind together with the help of one complex. That complex is called as synaptonymal complex. With the help of this complex, synaptonymal complex, both the homologous chromosome are remain in close contact with each other. This pairing, when they form pair, this is one chromosome, this is another chromosome. This pairing of chromosome is called as synapsis. And these two chromosomes, this is first chromosome, this is another chromosome. These two chromosomes are called as bivalents. This pairing is called as synapsis, and these two chromosomes are called as bivalents. Then, what about centrosome? Centrosome, which is already migrated, it is on the way of migration, it is moving only, it continues with its migration and it starts moving, or it continues to move towards the opposite pole. So here we have completed about this second stage of prophase 1, that is zygote. Now coming to pachytene. Pachytene is the most important step because here the two steps are taking place. First is the chromosome get undergo crossing over or overlapping takes place, and second important thing is exchange of genetic material. So the pachytene is the most important step. Okay, now see what happens. Suppose these are the homologous chromosome. Now these homologous chromosome, and these are called as chromatids, non-sister chromatid. Chromatid of one chromosome is called as sister chromatid. But the one chromatid of this chromosome, one chromatid of this chromosome, so these are called as non-sister chromatid. What happens in pachytene? The non-sister chromatid undergo overlapping. It continuously overlaps. It undergoes crossing over at many places. And what is the aim? Finally, what they do? They exchange their genetic material. Can you see in this diagram? This yellow color part came here and this blue color part come here. Okay, they exchange the genetic material. So what important thing is taking place in this pachytene? The homologous chromosome undergo overlapping, the non-sister chromatids undergo overlapping at many places, that is called as crossing over, and because of this crossing over the genetic material get exchanged. These two chromatids exchange their genetic material, and because of this recombination takes place. Then what about centrosome? Centrosome already they are on the way of migration, so they continues to be migrate towards the opposite pole. So here we have completed pachytene. Now coming to diplotin. What happens in diplotin? Now the aim is over. The exchange of genetic material took place. Now the chromosome, now after completing their aim, after exchanging their genetic material, they start migrating or they start repelling each other. They move away from each other, but at a glance only they cannot get separated. So slowly they start repelling each other. And the point at which they are still in contact with each other, immediately they cannot get separate from each other. The uncoiling takes place slowly, the repelling takes place slowly, and now few points are there where they are in still contact with each other. That points are called as chiasmata, that is called as chiasma, or it is also called as chiasmata. Now, what about centrosome? Centrosome again continues and it is just near to the pole, opposite pole. Okay, so this we have completed about diplotin. Now, the last step of prophase 1 is dikinesis. Now, see what happens in dikinesis. In dikinesis, again the repelling continues and it comes near to terminalization or termination. So what happens here? The chromosomes get separated from each other. See, the chromosomes, they get separated from each other, but only at the end. Only at one point they are in contact with each other. That's why this position is called as terminalization. Terminalization means now the overlapping is going to terminate. Now the attachment is going to terminate. Both the chromosomes will get repelled from each other. Coming to centrosome. Now the centrosome reaches to the opposite pole, and what is their aim? What they are going to do from here? They give rise to spindle fibers. They give rise to spindle fibers. Now when there will be the spindle fiber formation, there it may take place that because of this nuclear membrane and nucleolus, the spindle formation will not take place properly. So in order to avoid the disturbance caused by the nuclear membrane and nucleolus, the nuclear membrane and nucleolus automatically starts disappearing, so that the spindle formation can take place properly. Okay, so here we have completed dikinesis. In dikinesis, they are separating from each other. Only at one end they are attached. That is called as terminalization. Centrosome reaches to the opposite poles and it gives rise to spindle fibers. Nuclear membrane and nucleolus, in order to not create any disturbance, they get disappear. So here we have completed prophase 1. Now coming to metaphase. Now what happens in metaphase? M for meta and M for middle. What happens here? The chromosomes arrange themselves on the middle or on the equator of the cell. See, no nuclear membrane, no nucleus, and the chromosome arrange themselves on the equator or at the center of the cell. Then, see, nuclear membrane and nucleolus completely disappear. What about centrosome? Centrosome gives rise to two types of fibers. One fiber. Can you see from the diagram, this fiber which is arising from the centrosome and coming up to this chromosome. The fiber which is arising from centrosome and coming up to chromosome. This type of fiber is called as chromosomal fiber. And another type of fiber, the fiber which is arising from centrosome, that is from one pole and reaching up to the another pole, that type of fiber is called as interpolar fiber. What happens in metaphase? Here the chromosomes arrange themselves on the equator or on the middle of the cell, and nuclear membrane nucleolus completely disappear, and the centrosome forms two types of fiber. One is called as chromosomal fiber, another one is called as interpolar fiber. Chromosomal fiber means the fiber which arises from centrosome or from one pole and reaching up to chromosome is called as chromosomal fiber. Another is called as interpolar fiber, means the fiber which arises from one pole to another pole, that is from one centrosome to another centrosome, is called as interpolar fiber. So here we have completed metaphase. Now coming to anaphase. Now what happens in anaphase? Because of continuous contraction of this chromosomal fiber. Chromosomal fiber are these which are started from centrosome and reaching up to the chromosome. Because of this continuous contraction of this chromosomal fiber, what happens? The half number of chromosome. Suppose 46 chromosomes are there, 23, half number of chromosomes means 23 chromosomes get pulled towards one pole and remaining 23 get pulled towards another pole. What happens in anaphase? In anaphase, if total 46 chromosomes are there, half number of chromosomes get pulled because of contraction of chromosomal fiber, half number of chromosomes get pulled towards one pole and remaining half number get pulled towards another pole. This is only happening in anaphase. Coming to telophase. Now what happens in telophase? As we have already studied in anaphase, half number of chromosome gathered towards one pole, half number of getting shift towards the another pole. What happens in this phase? In telophase, half number of chromosome means 23 chromosome get gathered in at one pole and 23 chromosomes gathered at another pole. First, then what happens? Nuclear membrane. Now there is gathering of half of chromosome. Now around this chromosome, nuclear membrane reappears. Around each and every group which is present on both the poles, around each and every group, nuclear membrane reappears. Now inside the nuclear membrane, nucleolus develops or nucleolus reappears. So in this way, can you see, from one nucleus, two nuclei are formed in a single cell. Two daughter nuclei are formed, that is karyokinesis took place. Then this new spindle fibers, now there is no use of spindle fiber, so spindle fibers there only it get degenerated, okay, because the work of spindle fiber is over. To just pull the chromosome towards the opposite pole, that is over. So spindle fiber is completely disappearing here in telophase. So here we have completed about telophase 1. Now, karyokinesis over. Karyokinesis means division of nucleus, that is over. From one nucleus, two nuclei are formed. Now, coming to cytokinesis. Now what happens in cytokinesis? See, cytokinesis means division of cytoplasm. How karyokinesis? Karyokinesis means division of nucleus. Now we are studying cytokinesis. Cytokinesis means division of cytoplasm. Division of cytoplasm in animal cell and in plant cell is different. See how it is different. What happens in animal cell? In animal cell, when there is formation of two daughter nuclei, when karyokinesis took place, now it is going to followed by cytokinesis. So what happens? The notch or the furrow development takes place in cell wall. Notch development takes place here. This notch proceeds from periphery towards the center. This notch development proceeds and finally the whole cytoplasm get divided and it results into the formation of two daughter cell. Means here from one cell cytokinesis is taking place, division of cytoplasm is taking place. But how it is taking place? Furrow or notch development takes place in the cell wall. This furrow or notch deepens down and down at the center and finally it results into the separation of two daughter cell. While the case is different in plant cell. In plant cell no notch development or furrowing is take place here. But actually what happens here? Here the hard cell plate formation takes place between two nuclei. These are two daughter nuclei. Cytoplasmic division should take place. So what happens? How the cytoplasm is going to divide? Here in between the two nuclei, hard cell plate formation takes place at the center, which proceeds towards the periphery. From center it proceeds towards the periphery and finally it results into the formation of two daughter cell. So in animal cell, furrow development takes place from periphery to center and results into the formation of two daughter cell. While in animal cell, while in plant cell, cell plate formation takes place in between two daughter cells, two daughter nuclei, which proceeds from center towards the periphery and finally it results into the formation of two daughter cell. So in this way cytokinesis took place and from one cell two daughter cells are formed. Now, see, this is the just brief idea about meiosis 1 and meiosis 2. Now see, this is all about meiosis 1 and this is all about meiosis 2. Meiosis 1 is quite lengthy process. It is completed in 5 phases: liptotene, zygotene, pachytene, diplotin, dikinesis, as we have discussed now. Now, metaphase 1, anaphase 1, telophase 1. Now, meiosis 2. Meiosis 2 means from one cell, now two cells are formed. Now, what happens in meiosis 2? Meiosis 2 is again divided into 4 phases. That is prophase 2, metaphase 2, anaphase 2, telophase 2. It is just similar to mitosis. What happens in prophase 1? In both the cell, from one cell, two cells are formed. In both the cells, chromatin network get condensed. Okay. Centrosome starts migrating towards the opposite pole. Chromosomes appears to be long and thin here. And nuclear membrane and nucleolus starts disappearing. Same as mitosis. Prophase of mitosis. Now, coming to metaphase. Now, see what happens. Nuclear membrane and nucleolus completely disappear from here. Metaphase 2. Then, M for meta, N for middle. What happens here? The centrosome reaches towards the opposite pole. Okay, they give rise to spindle fiber, and the chromosomes arrange themselves on the equator. Only two events are taking place. In metaphase, chromosomes arrange themselves on the equator and there is formation of two types of fiber. One fiber which starts from one pole to another pole is called as interpolar fiber. The fiber which arises from centrosome and reaching up to this chromosome is called as chromosomal fiber. Now what happens? In anaphase, suppose this is chromosome. Okay, this is centromere. What happens in anaphase? The centromere divides. Centromere of the chromosome divides and results into the formation of daughter chromosome. It results into the formation of daughter chromosome. Now what happens here? Here, if these 3 chromosomes are there, it divides and it results into the formation of 6 daughter cell. And because of same thing is happening here also in another cell. from 3 chromosomes, 6 daughter chromosomes are formed. Now, because of continuous contraction and relaxation of chromosomal fiber, the half number of chromosomal fiber get pulled towards one pole, half number of chromosomal fiber get pulled towards another pole. This is happening in Anaphase. Now what happens in Telophase? The daughter chromosome get gathered at one pole. Here also daughter chromosome gathered at another pole. Same thing is happening here also. Now there is formation of nuclear membrane. Means from one nuclei, two nuclei are from one nucleus, two nuclei are formed. Okay, and nucleolus reappear over there. Now there is no use of spindle fibers, so spindle fibers disappear here. So in this way, karyokinesis took place, means from one nucleus, two nuclei are formed in both the nucleus, in both the cells. Then it is followed by cytokinesis. Cytokinesis means again the furrowing takes place and from one cell, two daughter cells are formed. Okay, so here in this way, meiosis 2 is also completed. Only one difference you have to keep in mind. In meiosis 1, the whole chromosome is getting, half number of whole chromosome is getting shifted to opposite pole. Means if 46 chromosomes are there, 23 are getting pulled towards one pole, 23 are getting pulled towards another pole. But here in Anaphase 2, there is formation of daughter cells. There is formation of daughter cell, means if 46 chromosomes are there, it will result into the formation of daughter cells, means 92 daughter chromosomes are formed and they will get pulled towards opposite, means 46 towards one side and 46 towards another side. So here we have completed meiosis 2.